I'll just do a quick introduction while we're waiting. So, hi, I'm Stephanie Halwa. I'm a PhD student at the University of Manchester. And hopefully, when the slides start working, I'm going to be talking to you about investigating the Apollo 16 regolith record in preparation for surface missions. And I'll try and keep it nice and short, as I'm sure we'd all rather be in the pub right now. And I'm having a bit of a coffee crash myself, so. Cool. Well, I'm sure most of you have seen this figure before, but it's a map of the lunar South Polar region. And the green-blue dots, which probably aren't showing up very well, are postulated exposures of regolith ice. And this is important because a lot of the upcoming missions to the moon all want to sample the abundance of these volatiles. One in particular is an experimental package led by ESA called Prospect. And this aims to measure the abundance and isotopic composition of hydrogen and other volatiles, including the noble gases. So the regolith in the proposed landing area is dominated by hyaline crustal material. And this is similar to Apollo 16. So in preparation for this mission, it's important to understand impact mixing processes in typical ice-free regolith and how this might affect the volatile budget. Oh yeah, sorry, I forgot to show that one. That's a picture of Prospect, which you've all heard about this week loads. So looking at the Apollo 16 regolith, we have three key goals. We want to determine the Apollo 16 Highland regolith exposure history and how it has been disturbed by impact events, define a dry Highland regolith volatile budget, and investigate the sources of volatiles in these samples. So Apollo 16 visited the central near-side lunar highlands, and you can see this in the red circle. And the samples we've chosen to look at are from double drive tubes, pictured here, which sample up to 60 centimeters depth in the regolith. I'll just show a quick site map. The zoomed in boxes show the locations of the double drive tubes. I don't know how this works. I'm not going to bother using the laser because I can't figure out. So at the bottom, we have station 4, which samples the Descartes formation. We have station 8, which samples South Ray ejector. And we have 2 at station 10 near the lunar module, which samples the Cayley formation. And also note next to these station 10 ones is a deep drill core, but we've not looked at samples from these. So we received polished thin sections from various depths from NASA JSC, and you can see a picture of this in the top right. And we've looked at their mineralogy using a software called ChemScan, which is connected to an SEM. And this produces mineral, element, and backscatter electron maps. And I'll talk through the stages of ChemScan in the next slide. But we then use these mineral maps to identify class of interest, which we looked at for their major element abundances using electron microscopy. We also received 50 milligram regolith subslits from the same core depths as the thin sections, which we've used for noble gas analysis, which can tell you about a regolith's exposure history, but also to look at the abundances and sources of volatiles. So noble gases are measured on a noble gas mass spectrometer. And after samples are loaded, we bake them for 24 hours at 200 degrees C to basically liberate any surface adhered terrestrial contamination. And so then the samples are heated using a diode laser to liberate the gas that we want to measure. And we use 4 laser heating steps ranging in power from 0.01 to 21 watts, which is around 600 to 1300 degrees C. Oh God, that's not come out very well. So ChemScan is a software that assigns a mineral to a pixel or a spot that's 5 microns in size. And it uses the BSE brightness and the EDS spectra from this point and compares these values with the values of minerals, which are all defined in this list, which is called a SIP list or a species identification protocol list. And basically the minerals are all defined by the colors, sorry. And we get these beautiful mineral maps as shown on the right hand here. And from these mineral maps, we can work out phase abundances in the thin sections. So here we have a histogram of the phase abundances. On the x-axis, we have all our samples. The depth through the cores go left to right, and we have each core in the bigger writing. And as expected, Apollo 16 is made up dominantly of feldspathic phases, but there is a lot of variation with depth, especially with the mafic minerals. And also there's no correlation between different stations, which makes it quite hard to map ejecta layers, as been previously stated in the literature. Two samples of interest are these ones highlighted here, and they contain exotic clasts, which I'll talk about in the next few slides. So the first one is from station 4. It's a sample from 42 centimeters depth, and this contains a Mare basalt clast, which is shown here on the bottom right. We have a plot of pyroxene iron versus titanium abundance. You can't see it very well, but if you look at the lovely screens over here, in the yellow, we have Apollo high titanium Mare basalts. In the light green, we have low titanium Mare basalts. And in the light purple, we have Crete basalts. And on top of this, the purple and pink squares are previous Mare class found from Apollo 16 samples. And the data from this study is the red and blue circles. And what you should be able to see is that they plot very nicely in the high titanium Mare basalt field, which is great. And they also overlap with the, I think it's Neal and Fagan values from 60639, which was a Mare basalt class proposed to be from Mare tranquillitatis. And so the other class is from a station 10 sample from 53 centimeters depth. We have a plagioclase anorthite number versus mafic mineral magnesium number. And you can see our data is the pink diamond, which plots very nicely in the fan suite. And you can just see a picture of this class here. So we can take ChemScan one step further, and from the mineral images, we can extract individual grains to have their own images. And so we do this by defining them as a group of pixels that have no pixels touching their boundaries. And before we separate them, we make sure we fill in voids in grains like agglutinates so they don't get split into several grains. And then we apply a separator process. And then we remove these manually added epoxy pixels, and we also reject grains that are less than 9 pixels in size, as we can't confidently identify them as grains. And so once they've been separated out, we can look at the individual mineralogy of all these clasts and their phase abundances, and then group them into lunar grain types based on their phase abundances, and we can separate agglutinates from glass and breccias based on their void abundances. So here we have some agglutinate abundances to look at regolith maturity, which is a measure of a sample's exposure to space weathering. On our x-axis, we have our agglutinate abundance. On the y-axis, we have depth. And on the upper x-axis, we have the ISFEO index, which is plotted in the sort of purpley line. And that's from literature data. That's not our data. But what is our data is the blue line with the stars. So these are the agglutinate counts from ChemScan. And the green and yellow lines are previously optical counts of agglutinates from the literature. And what you should hopefully be able to see is that our agglutinate counts correlate really well with the literature, which is great because it means we can use them to look at stratigraphic horizons in the regolith. And so the red upper arrow is the depth of the uppermost superficial reworked layer, which is around 10 to 13 centimeters across the landing site. And the other black arrows are other stratigraphic horizons that are labeled. But I think what's most interesting is this core here, which shows some discrepancy with the literature. So the literature shows there might be a stratigraphic horizon at 47 centimeters, but this isn't shown in our agglutinate counts. And this begs the question of, is this a bias of the ChemScan method, or is this to do with heterogeneities on a centimeter scale? And so to answer this question, we've looked at noble gases, and I'm just going to show you a snippet of the noble gases because this is really recent work. But on the left, we have argon 36, which is implanted by the solar wind. And on the right, we have argon 38, which is cosmogenically produced. So both of these are a basic indicator of a sample's exposure. And both of these increase with longer exposure times. And so we have a log scale on the x-axis for concentration and depth on the y-axis again. Our data is the green line, and it's the only study on this double drive tube. And then the other drive tube from station 10 has some data, which is in the purple line, and the deep drill core also has some data, which is the blue line, and Apollo 16 surface soils are plotted along the x-axis just for reference. But what I'd like you to take away from this image is that the noble gases, again, are very homogeneous with depth, which indicates this core is relatively well mixed and correlates very well with the agglutinate abundances that we saw. And this is also very important for Prospect as it aims to measure noble gas abundances with samples from different core depths. I don't know if you remember that drill. And then we can compare this with hopefully wet samples that should show, fingers crossed, elevated concentrations. So just a quick overview. Stratigraphic horizons vary across the landing site with no local correlation. There is an abundance of exotic material from non-proximal regions. The depth of the uppermost reworked layer is around 10 to 13 centimeters across the landing site. I don't know if you noticed at Station 8, it didn't contain this, but there's a theory that the surface layer was removed recently by an impact event, which is why it wasn't on that one. Argon isotopic concentrations and agglutinate abundances indicate the station 10 may be more homogeneous and well mixed than previously thought. Oh dear, I turned it off. And stratigraphic correlation may be possible using cosmic ray exposure ages. And so it's important to understand the mineral relationships with regolith disturbances, which can be correlated with the noble gas inventory in order to understand the regolith history in the lunar highlands. And this dry highland regolith volatile budget can be used to be compared with wet samples to be analysed on upcoming missions to the South Polar region, such as ESA's Prospect. Thank you for listening.
No, no questions is there? Yeah, I might ask one, even though. What are the next steps?
I mean, I'm sure you already know as my supervisor. But hopefully, so we've only been able to get argon, krypton, and xenon abundances, but hopefully it's to do with an issue we've been having with our noble gas mass spectrometer, but hopefully it will get fixed soon, so then we can get some neon and helium. And then I'm also going to look at getting noble gas data from two of the other cores, so that we have a range of samples to compare with Prospect. And then we'd also like to look at the hydrogen and nitrogen isotopic signature in agglutinates and Highland-derived impact glass beads, as there's not a lot of data on this, and it could be quite interesting to see what the trends are with depth and whether we have that, I guess, indigenous glass volatile signature with depth. But yeah, thank you.